Cleaning validation is a documented process that demonstrates our cleaning methods consistently remove residues to safe and acceptable levels. The validation verifies that our cleaning procedure can consistently and significantly reduce the amount of active ingredients, excipients, and cleaning agents to a concentration within the acceptance limit. In this video, we will explore aspects of cleaning validation, the difference between cleaning validation and verification, the steps involved in cleaning validation. Why is cleaning validation required? During the manufacture of pharmaceutical products, the surface of the equipment comes in direct contact with therapeutic ingredients and many excipients used in the formulation. Also, cleaning and sanitizing agents are considered as contaminants for future batches. The cleaning validation creates documented evidence that the cleaning procedure will consistently eliminate traces of residues from earlier products. What is the difference between cleaning validation and cleaning verification? Cleaning validation generate evidence for approving a cleaning procedure before using it. The validation activities include selecting cleaning agents, establishing sampling procedures, setting acceptance criteria, testing, and recording results. Cleaning validation is not a routine procedure. This is conducted for specific circumstances, such as introducing a new product changeovers, or equipment upgrades, etc. Cleaning verification, on the other hand, is a routine process that confirms the residues are removed from the equipment after each batch production. Unlike cleaning validation, cleaning verification is not concerned with the overall effectiveness of the entire cleaning procedure, but instead focuses on confirming the cleanliness of a specific batch. Types of cleaning processes: manual cleaning and automated cleaning. 1. Manual cleaning. Non-dedicated equipment such as mixing vessels, granulators, blenders, dryers, etc., is cleaned manually after each process. These are smaller in size and easily portable between processing areas. Tablet compression machines, filling machines, homogenizers, fluid bed dryers, and other equipment are also cleaned using manual cleaning procedures. Purified and distilled water is primarily used for cleaning where the active ingredients and excipients are soluble in water. Other cleaning agents are also used if the residues are partly or fully insoluble in water. 2. Automatic cleaning in place, CIP. Large processing equipment such as sterilization tanks, mixing tanks, storage vessels, etc., is equipped with an automatic cleaning in place, CIP facility. CIP is the procedure by which flush and rinse solutions are sprayed onto all internal product surfaces, without the intervention of operators, and continuously recirculated or flushed to drain for a predetermined time. Most CIP applications will use fresh distilled water discharged through fixed spray balls, which provide complete coverage of the inside of vessels. Types of cleaning agents in cleaning validation. Cleaning agents, such as detergents and other chemical aids, should only be used when cleaning with water or solvents is inadequate. Only approved cleaning agents should be used whose compositions are known. This allows analytical measurement of residues and has proven to be easily removable. Sometimes it is necessary to use sanitizing agents as part of cleaning, such as 70% alcohol, quaternary ammonium compounds, phenolics, sodium hydroxide solution, etc. How to perform the cleaning validation step by step. Step 1. Identify process, equipment, and product type. Identify all equipment that comes into direct contact with pharmaceutical products. This typically includes large and small tanks, vessels, pipework, blenders, granulators, tablet presses, transfer lines, etc. Equipment that is used for multiple products, has a complex internal structure, or cannot be fully dismantled, is prioritized for cleaning validation. Step 2. Worst-case product selection, solubility and toxicity. Perform a risk assessment to identify an indicator drug product. The indicator concept enables products to be grouped based on similar formulations, active ingredients, and shared production equipment. Identify the worst-case product based on your assessment of solubility rating and toxicity. To ensure the cleaning procedures are robust, always plan for the worst-case scenario. This means selecting the product that is the hardest to clean, the most toxic, or the most potent. For multi-product equipment, it is not practical to validate the cleaning of all products that share a single cleaning process and are similar in formulation and dosage form. Sometimes multiple products are grouped into families, and the worst-case product represents the entire group. This approach streamlines validation while ensuring it still covers the highest risk. Step 3. Select the cleaning procedure. The cleaning procedure is a step-by-step manual that explains exactly how each piece of equipment must be cleaned after production. Equipment cleaning involves dismantling, selecting appropriate cleaning agents, specifying concentrations, contact times, temperatures, and rinse cycles. It also outlines how to inspect equipment visually during cleaning and how to handle cleaning accessories, such as brushes or spray balls. Well-written, practical cleaning instructions reduce the risk of human error and ensure repeatable results. Step 4. Determine sampling procedure. After cleaning, you need to confirm that residues have been removed through cleaning verification. Appropriate sampling techniques must be used for adequate cleaning verification. The two primary sampling methods are swab sampling and rinse sampling. Swab sampling involves wiping a defined surface area with a sterile swab to collect any remaining residue. Rinse sampling involves collecting the final rinse water that has passed through the equipment surfaces. Critical sampling points are chosen based on risk, such as areas that are hardest to clean, including vents, gaskets, transfer switch, dead legs, valves, corners, and spray nozzles. A sound sampling plan defines the number of samples to take, where to collect them, and the technique to use. A successful cleaning validation can prove that the sampling technique, including sampling location and frequency, is justified for appropriate cleaning. Step 5. Validated analytical methods. After cleaning, it is time to check if the cleaning procedure has done the job correctly. For this purpose, the collected samples are tested using validated analytical methods. The methods must be specific to identify the residue, whether it's the active ingredient, excipients, detergents, or microbial in nature. Analytical methods must be sensitive enough to detect trace amounts, often down to parts per million, ppm, or even parts per billion, ppb. This ensures no harmful levels of residue remain. Examples of common methods include high-performance liquid chromatography, HPLC, total organic carbon, TOC analysis, and microbiological tests for bioburden. Validation of these methods is a mandatory GLP requirement. The method's accuracy, precision, specificity, and detection limits must be demonstrated so we can trust the results every time. Step 6. Establishment of acceptance criteria. You have two options while calculating acceptance criteria for rinse and swab samples. You should apply the most stringent of the two. Step 7. Cleaning validation protocol execution. The cleaning validation protocol consolidates all the elements into a single, controlled plan. It specifies which equipment and products are covered, who is responsible for each step, and what data must be collected. It details the cleaning procedure, sampling plan, analytical methods, acceptance criteria, and the number of successful validation runs required. Typically, 3 consecutive successful runs are required to prove consistency. All steps must be followed exactly as written. Any deviations must be documented, justified, and assessed for impact on the validation outcome. Once the protocol is executed, the results are compiled into a final report, reviewed, and formally approved by the responsible departments. If you have any questions or would like to learn more about this topic, please visit our website, www.gmp-SOP.com. You can find free, in-depth information on cleaning validation in the pharmaceutical industry on our blog. Please see the article link in the description below. Subscribe with us to gain access to a library of hundreds of well-written GMP documents. Use the link in the description to reach us.